couldn't find.
Welcome to Nusantara, the Southeast Asian archipelago. It is the birthplace of humanity, in our long journey of civilization. A homeland for human ancestors, for the last 1.5 million years. A place where our early ancestors have roamed a vast land of opportunity. Shared the space with many other wonderful creatures. Leave us the evidence of human evolution and their adaptation to the environment. Who are they? What do they look like? And how do they live their life? For almost 2 decades, the Center for Prehistory and Austronesian Studies has contributed to revealing the answers. Behind CPAS is a group of passionate scientists with global networking in prehistory and Austronesian studies. Our mission is to provide best practices in the framework of archaeological research and development. Pushing the values of our research beyond the knowledge. Reveals the national identity. Enriches our understanding of cultural developments. Unveil the reason behind our strong character of diversity. Narrates the story of ethnogenesis and the foundation of our nation-state. Preserving our past as a heritage for the wealth of our future generation.
Selamat pagi, good morning everybody. Thank you for joining us for today's lecture titled The Antiquity of Malaria and Thalassemia in the Mainland and the Island of Southeast Asia. So to shorten our time for today, because we already begin quite late, I will introduce, don't worry Patuman, I will introduce to Professor Truman Simanjuntak as the head of CPAS today, and he will deliver, as usual, the introduction to our today's topic about the antiquity of malaria and thalassemia in Southeast Asia. Please, Patuman, the time is yours.
Okay, thank you very much, Ruli. Thanks for all, Melandri, Mas Sofwan, and some others, all of the participants. So, selamat pagi, good morning for everyone. Yeah, this is our the fourth second talk show that we have already organizing up to now. So it has been more than 3 years. We organized a talk show and regularly every month. And today we have the special topic, and with the speaker, our best colleague, yeah, Melandri Forok. I think all of us know who is she. Yeah, he is I can say still a young researcher and very, very dynamic, very productive in doing research and publication. And our discussion for today is in the background of our collaboration. So we have three parties of collaboration between the Center for Prehistory and Austronesian Studies and the BRIN. Apa bahasa Inggrisnya BRIN? What is the English of BRIN? Okay, let's see in the
National
dictionary
Research
later.
and Innovation Agency, Prof.
Innovation and etc., etc. And also with our colleague, Dr. Melandri Fogg, yeah, Melandri Fogg. Yeah, so this will be, I think, will be a very interesting topic because so far we haven't yet discussed about ancient pathology or prehistoric pathology. And today we have the new topic titled is Antiquity of Thalassemia and Malaria in Mainland and Island Southeast Asia. Yeah, so it will be a very interesting topic, I think. And this is so very surprised when the collaboration team several months ago, yeah, did analyze of the human remains from Gua Harimau in South Sumatra. So I heard from Melandri and other colleagues that this team found surprise data. Yeah, so they identified the presence of the diseases that we haven't known before. Yeah, because before we know the disease from the dweller of Gua Harimau is limited to tuberculosis, caries, bruxism, etc., etc., but not yet known about malaria and also thalassemia. So this is a big surprise for me, and I think this collaboration is very prospective for the future. Yeah. So we know now there are many or different pathologies in prehistoric men in Indonesia. And the more interesting discussion because the discovery of from Gua Harimau is seen from the Southeast Asian perspective. Yeah. So I think this discussion will be broadening our knowledge about prehistoric pathology. Very, very interesting. So congratulations to our colleagues, yeah, our counterparts, especially Melandri and the teamwork who gave the new discovery. Just short information that that the Gua Harimau is a never-ending research due to the richness of its archaeological remains. So, as you know, so far there have been many important discoveries from this site. For instance, we have the long chronology of occupation from Upper Paleolithic to Pre-Neolithic, Neolithic, up to the Paleometallic periods. Not many caves that we have in Indonesia providing such complete chronology of prehistoric times. And don't forget, we have also discovered more than 80 complete human burials from this cave. And this is not, this does not include human remains found separately. Yeah, so we can imagine that this cave is very, very prolific for human remains. And also don't forget, if the excavation is still continued, I'm sure that there will be many, many more human burials found at this cave. And also don't forget that at this cave we have the first discovery of rock art in Sumatra Island. Yeah. And the least and very, very important is the discovery of the oldest metal artifacts from Harimau Cave. So we have the oldest date of the metal objects found in prehistoric times in Indonesia. That is from around 4th century BC. And it developed up to the 1st century AD. Very, very interesting. So thanks to the discovery of the teamwork of our collaboration, so it means that Harimau Cave is really, really very important. And on that basis, I strongly support the tripartite cooperation that has yielded this important results. So, well, for not take too long, let me end this introduction. And once again, thank you very, very much. So have a good discussion. And I give the time back to the moderator, Ruli Fauzi. Thank you very much.
Thank you very much, Prof. Truman, and nice to have you here with our colleagues. And as you mentioned, this talk show also as a part of publication for the dissemination of the the results of our collaboration between National Research and Innovation Agency (BRIN), Badan Riset dan Inovasi Nasional, Pusat Riset Archaeometry, dan Center for Prehistory and Austronesian Studies, serta University of Notre Dame, tempat Mel bekerja, affiliate, dan sampai kita akan lanjut ke acara intinya, yaitu presentasi dari Melandri. We will proceed to the presentation from Melandri vlog. Before begin the presentation, I would like to introduce you to Melandri vlog. Maybe some of you haven't met her, and she's a lecturer in Charles Sturt University in Australia, and she also affiliate as a lecturer also in the University of Notre Dame and researcher. And a couple of months ago, we made a collaboration. We made the analysis in the collection of Gua Harimau, and it is that will be related with the today's topics. It's about thalassemia and malaria during the prehistoric times and the antiquity of these two diseases in Southeast Asia. And Mel, good morning, Mel. Nice to see you again. And I saw a couple of your colleagues, and I would like to say hello also to a couple of colleagues. And thank you for coming. And then also the viewers in YouTube. And Mel, please, the time is yours to give us the short lecture and the talking about the topics today.
First of all, I just want to thank everybody for your wonderful introductions. That has been really unbelievably kind. Pak Truman is correct. I am still a very young researcher. I have been exceedingly lucky to have mentors who have guided me through this research since I was 19 years old. I was 19 when I went on my first field trip to the Philippines, and I fell in love with bioarchaeology in Southeast Asia. I wanted to be a forensic anthropologist before then. I wanted to be Dr. Temperance Brennan from the TV show Bones, and being in the ground excavating, I was excavating a jar burial, and I ran my hand down the side of a jar and I was talking away. As most people know me, if you have met me, I talk a lot. And I was talking away to a friend and I just stopped and I felt the hand impression of the potter who had made this jar burial. And for me, it just felt like time had just compressed across those 2000 years. And I thought to myself, that this is what I want to do for the rest of my life. And I'm so incredibly grateful and thankful for my collaborators who work with me and share the incredible knowledge and depth of understanding that they have. It is the best part of my job to be in the field. So I specifically thank Pak Truman, Pak Safwan, and Masruli for their incredible guidance with this work in Indonesia. Today, I'm going to contextualize Gua Harimau in terms of disease, which is a little bit different, I'm sure, to what you're used to. But I think it's very important to try to understand why we went to Gua Harimau to look for thalassemia and malaria in the first place. And is very exciting because today I get to present some of our preliminary findings. As Masruli and Patruman said, it was only a couple of months ago. It was in June, July. So this research is very preliminary, but also exceedingly exciting. So I can't wait for where we're going next. We're going back soon because uh, just to jump the gun a little bit, we found so much disease at this site that we didn't get through all of the assemblage and we have to go back. So this is my presentation on work that myself and my team have done actually in mainland and island Southeast Asia, and also the story of how we've gotten to where my research is, because those who came before me are also really important to this story. Okay, so this is the main research team of the part of the project that I will be talking about today. So Pak Turman, Pak Safwan, and Masruli are really important, obviously, have done amazing work at Gua Harimau. They've characterized the bioarchaeology, the dental health, and other pathological signatures that has led me to know that there is something there to go further and explore at Gua Harimau. My PhD student, Tiwi Yuwono, came with us as well. She is my right-hand woman, and she is so exceptionally talented and has just been a treasure in the field to get through this project. And that is the same for my project in Vietnam. So my right-hand woman in Vietnam is my PhD student, Minh Tran. And that was in association with Mark Oxenham, Hallie Buckley, and Kate Demet, who really don't need any introductions because they are the gods of bioarchaeology of Southeast Asia, just to put it frankly. So starting off, this journey actually starts when I am 3 years old. I was born in 1993. In 1996, this paper came out by now honorary associate professor Nancy Taylor from the University of Otago in New Zealand. And she noticed at this site known as Koh Phnom D, which is in southern Thailand, that the individuals had really, really thin bones. So if you have a look at the comparison here, the bones of these individuals are really thin, except for the fact that they also had really thick, some of them had these thick excessive new bone growth. So some of them had really thin bones and others had this really thick but porous bones. And the question was what exactly was going on. They also had really thick expansions of their cheeks. These are called your zygomatic bones, and also the forehead and the mandible as well. And so she determined that it was signatures of a very common genetic disease in mainland and island Southeast Asia known as thalassemia. Now, thalassemia is a genetic condition. It's one where if you get one gene, you have lesser or sometimes even asymptomatic signs. But you can also have severe forms as well. And if you have both genes, you have the most severe form. That's for beta thalassemia. In alpha thalassemia, you actually have 4 different genes associated with it. So you can have 1, 2, 3, or 4 of those genes inherited. Now, what this actually does is our red blood cells are made up of these proteins called hemoglobin. And each hemoglobin has 4 little chains attached to it, 2 alpha chains and 2 beta chains. So if you have alpha thalassemia, you have a defect in your alpha chains. If you have beta thalassemia, you have a defect in your beta chains. And what that does, it actually causes your red blood cells to die much earlier than they're supposed to. So you become really severely anemic. And as you become really severely anemic, your bone tries to adapt by producing more red bone marrow. But because it is genetic, you're producing more red bone marrow, but it's still producing defective red blood cells. So that means that you're constantly producing bone marrow that isn't producing proper red blood cells. And then your body wants to produce even more bone marrow because it wants to not be anemic. And you end up with these really, really thick expansions. So this is a child's skull. So this is actually bone, and in between that bone will be red bone marrow. Here you can see a common sign that we used to really not so great. We used to call it rodent faces, as in it looks like you've got the snout of a rat. But now we refer to it as what we call hyperplasia of the maxilla and mandible. So you end up with a really thick jaw and really thick cheeks, and it's actually your bones that are expanding on the inside. But they're also getting much thinner. So your bones are thick, but they're porous as well. As you can see that there's these big holes and there's red bone marrow that's filling into it. What's really interesting as well about thalassemia is when you are in utero, so you're in your mother's womb, your chains aren't actually 2 alphas and 2 betas. They are 2 alphas and 2 what we call fetal hemoglobin. But once you're born, those fetal hemoglobin get replaced with beta. So that means if you have alpha thalassemia, because you have always had alpha chains during your progression from a fetus to birth, you can have alpha thalassemia before you're even born. But beta thalassemia only starts to become symptomatic from about 3 months after birth. So there's about a 3-month time frame that we have in bioarchaeology to determine whether someone has alpha thalassemia or beta thalassemia. After 3 months, generally speaking, we can't tell the difference. But if you are less than 3 months old and you have these signs, we can say that the person has alpha thalassemia. So that is really important for some of the things I'll be talking about later. What is really important about thalassemia is that it has this relationship to malaria. So where malaria exists, thalassemia exists. And what is not on this map is where malaria has existed in the past. So malaria used to be in this area. It isn't anymore, but you still get the disease. Whereas in Africa and Asia, you still get the disease even where the malarial areas are. And in some places where it's either been eradicated or in some of these regions, we don't have a lot of data. There is overlapping between thalassemia and malaria. And here's why. So because the red blood cells die very easily, and they're also slightly misshapen, the malarial parasite actually can't infect the red blood cells that are damaged by thalassemia. So you end up having this thing that we call stabilizing selection, where most people in the population are what we call heterozygous. So they have one thalassemia gene and one non-thalassemia gene. So when they pass down to their offspring, they're passing down either thalassemia gene or a non-thalassemia gene. So it means that both the thalassemia and non-thalassemia genes are circulating in the population. And those who have the two genes, they typically die before they pass off their genes. Whereas those who don't have the thalassemia genes at all typically die off from malaria before they pass on their genes. So the highest level of people who have offspring generally are those who have both thalassemia and non-thalassemia genes. And we call this stabilizing selection. And we have a very strong association with malaria and thalassemia. So what do we know before we even look at the archaeology is, one, we know malaria is everywhere. We're talking about Asia today and Asia Pacific, so the tropics. Malaria is still a problem. It has been eradicated in lots of areas or eliminated in lots of areas. It has come back in some areas. In other areas, it never left. So it's a huge public health challenge in mainland and island Southeast Asia, and also South Asia as well, which is why it's so exceedingly common. Two, malaria has been around for a long time, and this is not based on studies that have been done in Asia, which contributes to the large research gap that we have. But we know that for at least 6,000 years, we have genetic clock evidence for malaria circulating in populations. Now, interestingly here, that is agriculture has already become a thing in Europe at this time. So the predominant literature talks about malaria being associated with agriculture. And as I'll talk about in a moment, that's not necessarily the case in Southeast Asia. Three, thalassemia has also been around for a really long time. And we know that because there's more than 800 variants of thalassemia and other what we call hemoglobinopathies, which are genetic diseases that affect the red blood cells. And they all provide some level of protective effect against malaria. So these 800 different types of mutations randomly happened in someone, but they happened to be in some sort of adaptive environment to spread and be very popular amongst the population. And that's where malaria seems to be the thing that has made thalassemia adaptive in Southeast Asia. And so there's over 800 spontaneous occasions where this mutation has emerged, and then it has spread and become really prevalent because it provides some level of protection from dying from malaria. So then the questions we are asking ourselves is, when did malaria originate in Asia Pacific? How did it end up in Asia Pacific? Where did it spread and when? So did it spread at the same time to mainland Southeast Asia as it did to island Southeast Asia? What routes, what migrations did it take? And how did this affect these ancient communities? And that's what we do as archaeologists. We're not just interested in the disease, we're interested in understanding the humans and the stories that exist in the archaeological past and how this really deadly disease has shaped the society and the culture of humans in our ancestral past. Here's the biggest challenge that we have, though, which is me as a paleopathologist who picks up a bone, looks at a bone, puts it down. I can't see specific changes to thalassemia. I can see things like really high mortality rates in children because malaria, especially the most deadly form, which is called falciparum malaria, it has a really high mortality rate for children under the age of 5. It causes significant growth stunting, so we can see generic markers of growth stunting in the bones. But all of these things can also be caused by lots of other things like hookworm, malnutrition, and all of these things typically come hand in hand. You know, you are more susceptible to malaria if you're malnourished. You're more likely to be malnourished if you have malaria. So it is really difficult to tease it out from traditional paleopathology methods. But if we're seeing in a sample really high levels of thalassemia, then maybe this is a proxy for malarial endemicity. So what I mean by that is if you've got high levels of thalassemia, then most likely you have high levels of malaria as well, because thalassemia is a deadly disease and it's not likely to be common in a population unless there's some sort of evolutionary pressure. So if you have maybe 1 or 2 cases of thalassemia, it's really difficult to determine whether that site itself had malaria because it could be spontaneous mutation, or it could even be someone migrating from somewhere else that had malaria and their ancestors were exposed to malaria, and then they move to a new place, and then you look at their bones. That doesn't necessarily mean that where you found their bones, there's malaria. But if you are working on a site where there are lots of people, children and adults that are showing this, then we have a really high probability that even though we can't directly see malaria, malaria is underneath all of this, causing or driving really high levels of thalassemia in the bones. I said before as well, so there are asymptomatic forms. So there are probably even more people in our assemblage who aren't going to show us clear signs of thalassemia who still had those thalassemic genes as well. So if you're finding something like 5 to 10% of the site has thalassemia, then actually there are more people in that assemblage that probably had it too. It's just that you're not being able to diagnose it very clearly. So the cases that I'm presenting to you today, I want you to think about that there are actually more people in this collection that likely had the trait. We are only able to see the really severe forms. So it's a signature for something much bigger in the assemblage. So that means because we have to take thalassemia into account, the question is, what is the antiquity of malaria? What is the antiquity of thalassemia? Most likely, the malaria would have had to come first because the malarial burden would have needed to be there already for a spontaneous mutation of thalassemia to become popular. So that means that we aren't able to necessarily determine the oldest cases of malaria through thalassemia, but we can at least determine when there have been cases that are really this malaria has been there and is established in the population. And then my last question is one that we want to use to be able to really establish even further beyond thalassemia, how old or how long malaria has been in Southeast Asia. And that's to try and innovate some approaches that we can use to go beyond the traditional paleopathology, which is what I learnt, where you pick up a bone, you look at a bone, and you record it. There's all sorts of new scientific techniques that we are looking at that we are interested in applying in the next 5 years or so as part of this broader project. So between 1996 and the next step, which I'll talk about in 2015, this theory developed about mainland Southeast Asia that wet rice agriculture, which doesn't happen until the late Bronze Iron Age, is what drove thalassemia. But the Neolithic in Southeast Asia is dry rice agriculture, and Kokpanomdi is a Neolithic site from 4 to 3,500 years ago. Iron Age is not until 2,500 years ago. So it doesn't really fit. The site Kokpanomdi, where we find this very clear evidence of thalassemia, doesn't actually match this idea that when we put moats down and we started irrigating to have wet rice agriculture, that's really when malaria got bad. The other question about this theory is that we actually don't have any evidence, especially around Thailand where this theory developed, in Iron Age sites that the malaria is causing really severe impact. So there's a problem with this idea. And when you think about where this idea comes from, it really is a copy and paste from the Mediterranean and African contexts. So the concept, which we call the first epidemiological transition or the agricultural transition, was that farmers arrived, slashed and burnt forests, and because of that, they left pools of water or stagnant pools of water which were beneficial to the larvae of the mosquitoes that pass malaria. And so that's what exposed humans, and that's why malaria has become endemic. That seems to be the case for the Mediterranean. It also seems to be the case in African contexts. So irrigation is highly associated with malarial burden in Africa. It is also seen in places in South Asia, like in Bangladesh. But the problem with this is that every single one of these mosquitoes have what we call different ecological niches. So each species and the genus that carries malaria, they're called Anopheles mosquitoes. Every single one of them, they occupy a different habitat space. So, and each one has a different potential to infect humans. So there's some that are really highly associated with infecting humans with malaria, and there are other species that are lesser associated. And in order to have malaria endemicity, you always have to have at least some of your humans in your group being infected by the mosquitoes that have the high burden malaria. We call them primary vectors. So what is very interesting is that the primary vectors in mainland Southeast Asia are called Anopheles dirus and Anopheles minimus, and they exist in forests. And you chase them away when you irrigate a site or you do slash and burning. So you actually remove them from the habitat by doing this. Other vectors do affect floodwater and moated sites and irrigation sites. These are called Anopheles barbarostris. They are called secondary vectors because they have a moderate to low chance of infecting humans. But even with that, there needs to be at least a small proportion of your people getting infected and then bringing the malaria into their community. And then in the bloodsuckers, the barbarostris, then pick up the malaria and keep spreading it around the community. So you will only have malaria spread by this one if malaria is also being spread by this one. So the model of the wet rice agriculture never actually fit, even before we started to get archaeological evidence which suggests against it. So then we jump to 2015, and this is my PhD supervisor, now Professor Emerita Hallie Buckley from the University of Otago in New Zealand. And she'd already been suspicious about this wet rice model and had spoken to a number of ecologists and also bug people, entomologists, who explained to her that that's not what happens in places like Vietnam and Thailand today. It's actually people who go on hunting and gathering um, at night or during the day in forests, and then they camp overnight before they come back to their village. They're the ones that are the most susceptible to malaria. And so she accompanied Mark Oxenham and Hirofumi Matsumura on their project on Konkono, which is a pre-Neolithic site. Sorry, that's not 46,000, 6,800 to 6,200 years ago. So seventh millennium. Here at this site called Konkono, a hunter-gatherer site, 2,000 years before agriculture becomes a thing in mainland Southeast Asia, and she noticed that the bones were really thick. But what's really interesting is that there isn't a lot of change on the outside because the bone has been remodeled. Now, this is a child. This is similar, but in an adult where it's already healed. And when they x-rayed, they could see what we call bone within bone, where there is a shadow of bone on the outside that overlies the original cortex. So this is actually this. It's just that this is someone in their 40s and it's healed. And if it wasn't for her skill to be able to think to look on the inside of the bone and not just the outside, we probably never would have known. As a professoress, she got very, very busy and parked the idea for a little while, sent some samples to our colleagues, including Dr. Justina Miskovitz, who then also worked with her then master's student, Meg Walker, on cutting some samples out of the bone and looking at it under a microscope, so doing histology, to see if we can investigate a little bit further about what is going on at the microscopic level. Then fast track to 2018, I contacted Hallie in the 2017 and started a PhD with her. And then in 2018, myself and my now PhD student, who worked alongside me as my research assistant, looked at the bones at Konkono again, and also looked at the bones from a very famous site called Munbak from the early Neolithic. Now, Munbak is 4,000 years old to 3,500, and Konkono is 6,800 to 6,200. Munbak is the same age, generally speaking, as those from Koh Phnom Deh, where Nancy Taylor had obviously gone and identified thalassemia. So I wanted to know if the, thalassemia was present in Munbak, because it's at the same time period. So is it more widespread during the Neolithic of mainland Southeast Asia? But also to investigate the suspicions that Hallie had looking at the thickness of those bones. And it really changed the game because Munbak, a whole bunch of these children had these like I said, we used to call it rodent faces, the really thick maxillar and mandible changes. They had really porous but large expanded skulls and faces and long bones and like shoulder bones, so scapula, pelvis, everything. So more than 10% of Munbak had these signs of really severe thalassemia. What was interesting was that some of them were surviving until childhood or even early adulthood. So there was a whole spectrum of impacts. But Konkono was so more poorly preserved. So Munbak was an excellently preserved site. That wasn't the case for thalassemia, so we had to rely on the histology. And the histology showed these really big pores, which happen on a larger scale here, which is that bone marrow expansion that's coming from the inside and pushing the bone marrow out and causing the bone to expand. So we were able to confirm that thalassemia was common in Manbak, at least infrequent, but definitely present in an important amount of people in Konkanor. So we argued that that meant that we have at least something like 7,000 years of malaria, and I mean malarial endemicity in Southeast Asia. Then in 2024, Ming and I went back, Ming this time as my PhD student, and we decided to do a top to toe analysis. We worked on, so we have now analyzed over 800 remains in Vietnam from sites that are reported to be 10,000 years old, right down to sites that were 1,500. We didn't find anything older than what was at Concanor, so Concanor still remains those oldest cases. But we found all these other sites down in South Vietnam and Central Vietnam that also had these same sort of challenging pathology of expanded bone where it was sometimes thin. In older individuals who had less severe conditions, it was thick, really thick skulls, expanded mandibles, and then sometimes you had enlarged what we call nutrient foramina, which is where your blood vessels go in and out of your bone. And if the red blood cells, the bone marrow underneath is expanding, it also expands the foramina as well. So we are now certain that from at least 7,000 years ago, thalassemia and there also malaria was very common in Vietnam from north to south for at least 5,000 years. By this time onwards, where we've got historical references to malaria, so what we have is a clear evidence of 7,000 years straight of constant malarial endemicity. So probably before 7,000 years, malaria was introduced, and it definitely became a big thing and a widespread thing very quickly in mainland Southeast Asia. But what about island Southeast Asia? Because that's what we're all here for, right? We know the story now relatively well in mainland Southeast Asia. We still want to do more work. We would like to eventually work in places like Laos and Myanmar and across the edges, Laos especially because, and Myanmar especially because of the Anopheles minimus is very focused in those areas. And also, we would like to go back to Thailand and look at some of those earlier sites that have been extensively studied. But because we know so much more about what it looks like, what thalassemia looks like in Southeast Asia, because it does look quite different to the thalassemia that is published for the Mediterranean. So now we have a really good understanding of what it looks like down here, associated with the mutations that have emerged down here in Southeast Asia. Now we want to go back to earlier sites and look again and do x-rays and all of those things because x-rays are very important for looking at the inside of the bone, whether it's been compromised from the expansion of the bone marrow. So that's where Gua Harimau comes in. Which, by the way, Professor Hirofumi Matsumura, I was in the field with him. I had just finished my undergraduate. It was March 2016, and he was telling me about this site, Gua Harimau, because I was thinking or choosing what sites I'm going to do on my PhD. We didn't end up getting there, but I had always in the back of my mind thought about Gua Harimau and just how badly I want to study this site because it looked amazing. And as Patruman said, you've got from pre-Neolithic to Metal period. Professor Hirofumi Matsumura's team, along with the Gua Harimau team, Patruman, Pasafwan, have published radiocarbon dates for some of the burials. and they're dated somewhere between 4,700 to 1,800 years ago. So that's a really long chronology where the climate is changing, so the dynamics or the patterns of the mosquitoes might be changing. What we do know now is this is right on the tropical belt. Just very recently, in the last 2 years, the health team in this district eliminated malaria, but Gua Harimau was actually the epicenter just about 3 years ago, we found out, for the malaria. And that's where they found lots of those, like I said, primary vector mosquitoes, which was in low riverways in the middle of the forest near Gua Harimau. So if it was there in recent times, was it there in ancient times? And so, yes, this is our wonderful team, and my PhD I am her PhD supervisor, my PhD student Tiwi, who will finish up in the middle of next year, and I don't even have time to talk about some of the amazing things that she has found, but there is a lot of disease in throughout other sites in Indonesia as well that I can say that Indonesia is there is so much potential for understanding what is happening in this whole region by studying Indonesia, and this is where Gua Harimau comes in. So the sites I was speaking about before, they're on the outer edges of the tropical belt. Gua Harimau is right there in the middle of that tropical belt, and just next door in Papua, we've got still really what we call holo endemic malaria. So up to 50% of people get infected with malaria in Papua. So this was definitely from a disease perspective and an environment perspective to go looking for evidence of thalassemia amongst other tropical diseases. So what I'm presenting today is thalassemia, but we have found so many other things at Gua Harimau. We are, like I said, we are going back because there are just so many exciting things that I don't even have time to talk about today. Yeah, so you will be hearing about this site, I think, for years to come. This is our team. So this project actually also involved collaborating with the local public health team. We are trying to come up with a community-led project where we're using some of the findings about Gua Harimau to tell stories of the people in the past as a way to educate about these diseases in the community today and to increase the disease awareness of some of the things. Now, I said that the team in around Batu Raja has eliminated malaria, which is exceptional. When we found that out, I couldn't, like, it was amazing. And really kudos to them for the amount of effort and that has gone into that. We want to make sure that it stays eliminated and that we can use that space as a way to work with other nearby districts who don't yet have elimination. So to prevent those nearby districts from importing malaria back into the area, but also to spread that education from an elimination area outwards so that we can keep extending that elimination zone to the benefit of the communities. So that's the purpose of this project, is to actually bring archaeology and modern public health together. It's very ambitious. This is just part of our team. An incredible team. We all wept and cried when we said goodbye at the airport, but also some incredible findings. So the first case I'm going to show you is a very clear case of thalassemia, most likely beta thalassemia, because there are some slight differences with beta thalassemia typically being much more prominent to the skull and to the face. So you can see on the x-ray just how swollen and even here how blown up the mandible is. And then if you have a look at the maxilla and the cheekbones, they're also really swollen, and yet there's all these little holes, these little pores. So this is an individual from the Neolithic, dated to at least 2,700 years ago. And you can also see on his x-rays that his bone is not particularly healthy, or her bone, sorry, is not particularly healthy. So, right from the get-go, we started from pre-Neolithic burials, of which there are only a few, and moved on. So by day 2, day 3, we were already starting to see clear evidence for thalassemia before we had even had an opportunity to do X-rays. X-rays are obviously very important for thalassemia because it starts from inside the bone. So if you've got effects on the outside, then likely the inside's going to be even worse. And you can see the extra margin that's around the teeth and all these extra bits of space, and that's that bone marrow. So if you put it together, you can see just how chunky the cheekbone is, and then how rounded the upper lip is, and upper jaw, and then lower jaw as well. So right from there, and then it didn't stop. So we have since identified about 4 cases of thalassemia in we've studied so far only 25 individuals. But what is even more exciting is these cases from the metal period. Now, at Munbuk, we had a baby that had possible case of thalassemia. So in that paper that was in Scientific Reports, we had argued that alpha thalassemia and beta thalassemia might be happening. But that baby didn't have very strong evidence for thalassemia. It was only a possible case. When we were working on cleaning the bones for this individual, who was a 17, 18-year-old woman, very brittle, and you can see on her x-rays how compromised her bone was. Her bone was so light, you had to be very, very careful. The work we had to do with cleaning her bones had to be so delicate because she was so thin. And here you can see this extra bone here. And when you x-ray it, you can actually see that there is a section of the bone where it extends from. And that is a very strongly diagnostic marker for thalassemia. They look like bone tumors, but they're not actually bone tumors. They go all across the inside of the ribs down, and they're very specific to where they are. And so it's actually the bone marrow that's pushing out and expanding, just like the picture that you saw in the skull. And then the bone marrow has since decomposed, but there would have been red bone marrow sitting here. She's got other signs of anemia as well, so quite a thick skull for someone who is a 17-year-old woman, and other aspects of porosities. As I was cleaning out her lower ribs and her pelvis, I found this baby that she was still carrying. The baby is about 38 weeks, so just about ready to be born, but was exactly in the position of pre-labor. So she unfortunately died before this baby was born. And this baby has really strong signs of thalassemia. So there's extra bone expansion of the ribs, which again is what we call strongly diagnostic for thalassemia. So very strong signatures of this condition in a child who, by the way, this rib is blown up, but it's this small. This is about 5 millimeters in size. This child was so little. And then also his long bones as well, or their long bones had all those extra expansions as well. And as you can see, really porous new bone. And if you have a very keen eye, you can see that the trabecula, so the mesh-like bone on the inside, are also quite severely compromised. So we have, for the first time ever, a direct case of a mother giving thalassemia to her baby in the archaeological record. And that's only possible because she was so brittle, so whoever had excavated her had made the smart decision not to completely excavate her at site, but to block lift her and bring it back to the lab where she could be more properly excavated. Her bones were so brittle. And through that, like I said, I was able to find the baby inside, who and obviously passed away with their mother, and she was just obviously a teenager herself. So already we're only halfway with the burials that exist, and as Patruman said, there still are so many more burials that are left in Gua Harimau that there are so many things that are ready to be discovered at this site, and I'm hoping that this research uh, kind of bursts work again. There's an incredible museum and an incredible team who works in the museum at Gua Harimau. There are just so many opportunities for work, for collaborative work, and to the benefit of Indonesia. Thank you. Um, because, like I said, a lot of our work has been done in mainland Southeast Asia, but Indonesia is really an area where we can imagine that these things are happening. So now we know thalassemia is in island Southeast Asia by at least the Neolithic. So then we have more questions. Did they bring it with them when they migrated from Taiwan down? Or how was it there for longer? So did the indigenous communities have it? All these questions that have now been opened up because of these samples. But what is clear is once it arrives by the Neolithic, it's definitely carried on. These genes are existing in really high quantities throughout the Metal Period for the rest of the time period of Gua Harimau. So Gua Harimau Part 2 is coming soon. We're going to go back. So yeah, like I said, we've only looked at something like 25 individuals. 25 burials, sorry. There are more individuals to look at. There's a lot of careful, slow, painstaking work that we need to do because the bone is so brittle, and cleaning the bones very carefully. So, for example, making sure that these portions of extra bones stay there, and we treat the bones and these ancestors with respect, and we tell their story with the most respect that is possible. Because if it wasn't for them, we wouldn't have this knowledge. So what does that future work look like? Obviously, this work is continuing in Indonesia. Tiwi is doing incredible work at another site in Indonesia, along with her research partner who leads the site, Stuart Hawkins. And Mintran is also doing a lot of work in Vietnam, some characterizing what we can do to identify thalassemia in really fragmented remains using quantitative methods. We're going to do some mortality studies, so we're going to look in further detail on the growth stunting and all of that, and combine that with GIS epidemiology. We are wanting to map where the mosquitoes were in the past, and I'm working with a team at Cambridge University with that. We want to go back to Thailand, like I said, also the Philippines. As you can imagine, if it's in Indonesia, then the Philippines and Malaysia are also places that we need to go looking for this disease and other tropical diseases. But what is really, really exciting is the emerging field of metabolomics and proteomics, but especially metabolomics. Metabolomics is a way to identify byproducts or little chemicals that are produced or molecules produced in the process of the body responding to infection, and also the parasite responding to the body. So both the parasite is producing certain chemicals and the body is producing certain chemicals during active infection. So we have a team that is setting up a omics lab that's combining the powers of Charles Sturt University, of UNSW, so University of New South Wales, and also Leicester University, to come up with multi-omic approaches that we can use in the tropics. We're starting with other diseases first, like leprosy and tuberculosis, and then we'll be moving on to things like malaria, in hopes that we can identify malaria directly from the bone, and also the impacts that thalassemia can have as certain products, byproducts, chemicals are produced by the thalassemia as well. So thank you so much. Sorry, that's melandryblock.com. That's my email. And thank you to Gua Harimau Museum and also all the museums in Vietnam who were so kind enough to let us do this work. This is my amazing team. There's Masruli right there, who's essential to the team. And I cannot thank this team enough for the incredible hard work that went into this. Thank you so much.
Thank you so much, Mel. Great presentation. Remind me about our hard work in Gua Harimau. And also about the excavation. I still remember that Prof. Truman explained to us that we have to do a micro excavation in the lab next time, but we have to carefully keep the data fresh for the future archaeologists. And it was like I don't know, it's like 10 years ago, I believe, more than 10 years ago. And then honestly, we as young researcher heard, I'm sorry, Prof. Truman's still here. We cannot believe that. How do we bring the complete feature of burial to the museum? Meanwhile, when we walk to the Harimau cave, we struggle because it's really hard. It's you experience that.
I struggled
with with the dirt road, and then it's like hundreds of kilos of burial feature, and we succeed to transport it to the museum. It was quite an experience with Prof. Truman. And we learned a lot. We learned a lot as his students during our mission in Gua Harimau. And still, like Prof. Truman said, it gives us knowledge, new knowledge, and it's just because the how we excavate it and how to deal with the remains in one site. And thank you for that. And thank you for to make this real life, to the real world, that what we have done in Gua Harimau, it's actually giving a new opportunity broaden the opportunity for the future research. And then we will proceed to next discussion. And please, if you have for the participant here, if you have any questions or comments, we inviting you to asking directly to Mel or writing in the chat. I can give translation to Mel, or you can write also in the YouTube channel for the comment if you have any comments on works what we have done in Gua Harimau and about the connection between thalassemia and malaria in Southeast Asia. But Mel, I just got like a question when you explaining about the malaria and thalassemia in a couple of slides before. And I also read about several articles since you explained about this connection of that. I'm getting interesting on it, and there's some lots of articles talking about the connection between alpha thalassemia, making like a kind of protective mechanism for people living in the endemic place of malaria. Do you have any, until now, do you have any evidence from the archaeological perspective about this opinion, Mel?
The only thing that we have is the fact that we are seeing this in malarial areas. So we'll have, the generic cases of maybe associated with malaria, like growth stunting and things like that. And the ecology, so the environment is perfect for malaria. But until we actually can directly see malaria in the bones, it's going to be really difficult to use archaeology. But as you know, with archaeology, archaeology can't work by itself without drawing from other fields. So with the thalassemia, alpha thalassemia is extremely common, much more common than beta thalassemia. Um, and there are parts of places like in Vietnam, northern Vietnam, where up to 70% of populations have the at least one thalassemia trait in endemic malaria areas. And it also seems to be the case in Papua, really high levels of alpha thalassemia. In mainland Southeast Asia, they have this thing called HPE disease, which is not a thalassemia, but it also affects the beta hemoglobin, and it becomes a problem when you also have beta thalassemia. So we have all of these weird extra mutations that are very strongly associated with where malaria is today, and where we can guess, well, infer from the environmental context and the archaeological context where malaria was in the past. We've got clinical evidence now, which has emerged in the last 5 years, that indicates that there is a protective effect. So we know this relationship exists, but we have yet to actually be able to diagnose that relationship in the archaeological literature, purely because it's so hard to identify malaria, and we're using thalassemia as a proxy to identify malaria. Yeah.
Yeah, it's a matter of lack of evidence. Like it's really hard to distinguish actually, especially about the growth of the bones and the like you say, the relation on the surface of the bones. When we work on the collection in Gua Harimau, I realized it's quite hard, and it needs like a really good eyes on it. And and also like what like you say, it's really clear, it's more clear to be seen in the under the x-ray, under the radiograph, about this quite evidence. But
Yeah.
for sure we cannot do it from to all bone, right? The x-ray. But
So the hard bit is identifying it macroscopically first so that you can take it to
yes.
X-ray. In a perfect world, we would just take every single bone and X-ray every single bone. I
Mm.
have had the opportunity to do that once, in Japan.
Mm.
I worked in the National Museum of Science and Nature, and they had an X-ray machine, and they gave me the X-ray machine for free for a week, and I just scanned every bone in existence. But that's not, yeah, that was a one-time thing in my entire career. So you need to have the skills to be able to identify it macroscopically, and then have the supporting evidence from the X-rays. But the X-rays are so important for identifying this disease, exceedingly important.
Yes. So we are still waiting for any question or any comments from the participant today. And also for I saw several colleagues from Mel. I saw Ming before. Hello, Ming. Mel talking lots about you, good things only. Nice to have you here, Ming. And regarding our work for in Gua Harimau, do you think that this is related with the geographical distribution of thalassemia and malaria? Do you think that we in Indonesia we have like very diverse environment? Like you have seen what kind of environment in the central of Indonesia, in Sumatra, wet every day, and then also in the center of Indonesia, in the Wallacea area, and also in the Papua, we have quite different geographical subdivision really of the ecology, and also microclimate. Do you think that it is possible that that this kind of difference can produce different characteristic of the malaria spreading and also the thalassemia in that sort of kind of different environment.
You are absolutely correct. So Gua Harimau is, as you know, in Sumatra rains a lot. So it's exactly where you would expect malaria because the more it rains, especially year round, the more chance the humans have to be exposed to the mosquito. We don't typically see malaria in the highlands, like in the very high highlands, typically because it is cooler and also the altitude. The mosquitoes don't like the altitude. The other thing is in urban environments, you don't get malaria either. So one thing that is very beneficial to us as archaeologists in terms of reconstructing the mosquitoes is they're very particular where they want to live. And so they're not like the mosquitoes, the tiger mosquitoes, which are the Aedes ones that spread dengue, which, by the way, a lot of our team members got dengue when we were in Guwahati. Really, I don't know if you even know the update, but a
No,
couple of
I
us came
haven't.
back to Australia and it turned out that half of us had dengue fever.
Oh yeah,
Um,
sorry.
So, well, sometimes if you go looking for tropical diseases, they find you. So, just a word of warning for those who work in the tropics looking for tropical diseases. But those mosquitoes, the Aedes mosquitoes, they're very robust. They can live in the cities, they can live everywhere, and that's why dengue is everywhere. Whereas malaria, the malaria mosquitoes are very picky about where they live. Which also means that we have the ability to sustainably uh, work on our how we interact with our environment and or how we control our habitats and our environment to keep mosquitoes away with malaria. And the more we find this kind of work in archaeology, the more we understand how this disease behaves, the more we can also understand sustainable ways today to try to prevent vulnerable communities from being exposed to malaria. But yes, it's not like dengue. It is very in very particular areas, typically lowland and very wet regions. And that's why with climate change, it's a bit of a worry because with climate change, it's getting warmer and wetter more in more regions. Already in Papua New Guinea, there is the alarm call has been sounded because there never used to be. There's no historical evidence for malaria in the highlands, and now there are highland regions where there is malaria because it has gotten warm enough and wet enough for the mosquito to want to breed in the ground or in the pools in the highlands now. Yeah. So it's a bit
Yeah,
scary. Yeah.
yeah, yeah. I agree with you, but yeah, the relation between the climate change and the spreading of disease to current present day. It's a interesting topic because we found, yeah, the fact is it's getting larger, the area of that covers the malaria and also dengue and other vector disease uh, distribution. And maybe Pak Sofwan, you or Pak Truman, you have any comments on our collaboration research project in Gua Harimau? If you have some, maybe Pak Sofwan or Prof. Truman.
Okay. So, Melandri, you have a very new interesting finds at Gua Harimau. And I think you and your teams should publish this very interesting discovery. You don't have to stop at Gua Harimau, you analyze it, and then you finish your work. You have to continue with your team. Yeah. And another one, I'm so interested that you mentioned that slash and burn agriculture increase malaria risk in Indonesia. You mean that the malaria has been already exist before Neolithic, before the agriculture?
Yep.
And do you have evidence for this disease before the Neolithic?
So, when it comes to island Southeast Asia, we have a very good understanding of where in the mainland. Like I said, it is the forest mosquitoes in the mainland. So when you slash and burn, you actually drive the mosquitoes away. So it doesn't increase your risk, it reduces your risk. Um, there are many more vectors in Indonesia that can cause malaria. So one of the next steps that we need to do is to do like what we did with the mainland and work out which mosquitoes are the main vectors, how do they work, so which habitats, and which people are more vulnerable. At Gua Harimau, those that were vulnerable were those that were walking around the forested area. And now around the Batu Raja region, actually, the health people were saying that it was miners for metal, heavy metal miners, who were spreading malaria because they were getting exposed as they were coming home from work from the mining regions. So it's people going out into the forests or the jungle and then coming back, and during that time they're being exposed to the mosquito. So I'm not entirely sure what slash and burn agriculture does in Indonesia, whether it makes it worse or whether it makes it better or whether it's not a big deal. I haven't really found that much about Sumatra regarding slash and burn. A lot of it was focused on the remote communities with the husbands being miners and then bringing it back from the mines to their communities. So I have lots more work still to do in trying to characterize what the relationship is between um, the habits of people and their exposure to the vector, because that is going to help us understand what people were doing in Gua Harimau. What activities were they doing to make them exposed to malaria is a big question that we now have to answer.
Thank you very much.
Thank you very much for the answers. And maybe from Pasofwan, you have some insights regarding our preliminary results regarding the
Yeah,
bioarchaeological aspects.
yeah. Thank you, Mas Ruli. Mel, yeah, it's very interesting. Do you think there is a difference, difference resistance between one population and other population? As in island Southeast Asia, we have at least two wave or more than two wave migration in this region. Is there any difference on the resistance of malaria or the potency of thalassemia
Mm-mm.
in different
Yeah. So it's very complicated because we've had, as you explained, so many migration waves, and the genetic work that has been done has been on modern populations. So we see resistance in Melanesian populations and we see resistance in Polynesian populations, including Polynesian populations who live in places like Tahiti and Hawaii where there isn't malaria, but their ancestors traveled through malarial zones. And there is also a hypothesis that maybe they were the ones that brought it in the first place during the Austronesian expansion, but we have no direct evidence for that. So the question then is it who is more resistant, or where does that resistance come from? We don't know. We just know that there are definitely different mutations that exist between Melanesian and Polynesian populations, but there's also overlapping mutations as well, so ones that they share. But this is what my hope is, if we can understand or to start to do the metabolomics, because DNA, as we know, is not very good. In the tropics, we struggle very much with DNA, and many of the thalassemic genes are deletions, so they don't exist. They've been deleted out. And with DNA, it's very difficult to tell the difference between degraded DNA. So it's not there because the DNA is degraded, or it's not there because it's a deletion mutation. But that deletion mutation causes thalassemia, and then that thalassemia causes chemical change in the body. So if we can capture the metabolites, then we should be able to technically be able to capture the deletion, even though the gene is no longer there. So that's why I'm looking forward to what's going to happen with metabolomics, because we can understand the interaction in archaeological skeletons of malaria and thalassemia together. So that's why I've kind of skipped the DNA, and I'm more interested in what we can learn from metabolomics. I think we're about 10 years away from doing that. But I think in order to get there in 10 years' time, we need to start to do the work now and start to train people up, and build the capacity inside of Southeast Asia so that we can do this kind of metabolomic work.
Thank you very much, Mel. And I think it's answered, Pasofwan, your curious questions
Yeah.
about the resistance. It's also my question actually, but I questioning it's just curiosity.
Question. Yeah.
Yeah.
I mean, that's the thing. The relationship with the migrations is what's very interesting here. Did they bring the thalassemic gene with them, or did it emerge here in Indonesia? We don't know yet. And so we have opened up so many more questions for us to further explore, which, yeah, there's easily 20 years worth of work to answer those questions.
Yeah, and how do you think about the future of paleoproteomic studies for advantage for this field of research on thalassemia and malaria?
I think lipidomic paleolipidomic, paleoproteomic, and paleometabolomic, I have a feeling that they're going to take over from genomic research, and they're going to become much more standard, especially in the tropics, because a lot of the lipids and proteins that we work with are hydrophobic. They survive better in the tropics than the DNA does. And they're far more abundant than the DNA, and they're downstream. They're what the DNA is doing to the body. So I have a feeling that there's a whole new world that's going to open up for us in the tropics. And so we got to jump on board and start training up in Southeast Asia so that we have a very strong scientific base that comes from Indonesia, from Vietnam, from Malaysia, and that we can collaborate and work and come up with this incredible story, because the migration story is so big. It goes beyond one country. It's across so many different countries. And we need so much data to be able to capture the complexity that is human migration. And then that's just talking about maybe one migration. As we know, there are multiple. The more studies we do, the more migrations we keep finding. So yeah, the story is incredibly complex, and that's why we all dedicate our whole lives to this, because the more we study, the more confusing it gets.
Yeah. And I think the question also related to the facility, Pasofwan. What about the facility in Indonesia? According to you, can we do the, like we said, lipidomics, metabolomics, and proteomics in Indonesia for the next at least 2 or 3 years after today? Or
Yeah,
Is it possible to do it in Indonesia?
Yeah, now we have a laboratory regarding cryogenomic, especially for structural biology. It's mainly they study on proteomic, not paleoproteomics. I think we can, in the future, we can switch a little bit this study, not only for the reason proteomic, protein, but also to, yeah, maybe to solve the problem from paleopathological problem from paleoproteomic. I think it's possible. And also DNA, we have also a DNA laboratory. We can switch a little bit, not only a bit, because they have different, of course, they have different standard operational, but we can enlarge the study not only for recent DNA studies, but also for ancient DNA studies. Yeah.
Yeah.
This is an opportunity.
Yeah, Mel. So if you have any students, or even here we have, I maybe if some participant are planning to do a PhD, Mel currently searching also bright PhD future students who wants to do the subject that we mentioned before, more to the biomolecular aspect of bioarchaeology. And it will be possible to do it in Indonesia also, ya Pak Sofwan, regarding the facility we have in the National Research and Innovation Agency. So it's a mutual collaborative program, and I really have I have to say that I have lebih percaya diri. I have the confidence that we can make it in Indonesia.
Yeah.
And we have one question from the chat in Indonesian. What kind of plasmodium in malaria can be found in the population of prehistory? Maybe if we elaborate, the question is, can we identify the plasmodium causing malaria from the rest of the ancient population from the prehistory? Is it
So
possible?
what we can infer is that falciparum is driving thalassemia, because Vivax only in very specific circumstances causes mortality. So the evolutionary pressure has to be a very strong one. So when we're referring to thalassemia, we are probably referring to its relationship with falciparum. But here's what's really interesting is that we don't really understand the mechanism as to how thalassemia protects from falciparum malaria. There are two hypotheses. The one is that it is a direct protective effect, so very similar to sickle cell anemia, where the the parasite is not able to use the oxygen in the body very well. The second one actually is, and this is in relation to some work that's been done on alpha thalassemia, is the idea that maybe alpha thalassemia makes you more susceptible actually to getting VVAX as an infant. And so by making you more susceptible to getting VVAX, you then have a cross immunity to falciparum, which kills you off. So there's another argument then that if you have thalassemia, you might actually also have falciparum and VVAX in the mix. Now, these are all hypotheses that have not got a lot of evidence. And again, this is maybe a question we can answer with some of the research that we're hoping to do to improve the actual direct identification of malaria in the archaeological past. And maybe we'll be able to help try and separate some of those challenges that clinical medicine has regarding the relationship between malaria and thalassemia. Ruli on mute.
Thank you very much, Mel, for the answer. And I believe that the next question, also the next challenges, we faced during our works in Gua Harimau, it's about the state of conservation, Mel, of the bones, and also the technique for the conservation, also the way we conserve the bones. And do you have any comments on regarding also the subject of the paleopathologists, also the future opportunity for us to do paleometabolomics and paleolipidomics, etc., in the biomolecular aspect of the human remains? What should we do as archaeologists? I think that's the most important question. What should we prepare, what to do, and what we should not do on the human remains to have a best opportunity to enlarge the spectrum of the future studies today?
So there's actually there was just last week a conference in Greece on bone diagenesis, and they were talking about some of the issues with consolidation of bones, because consolidation um, preserves the bone to look really fantastic, and that's actually great for someone like me who's a traditional paleopathologist, as I can see the bone and all of that. But there are some issues with consolidation potentially contaminating the samples themselves. So at the moment already, ancient DNA people try and stay away from consolidated bones, but we use it. I mean, it's used a lot in Southeast Asia. It's used in Vietnam as well. And so I think the question that we really have to have is it's a balance between what are we conserving? Because your one scientist will say, I want the bone to be conserved in this way for this reason. And then you'll have another scientist asking, I want the bone to be conserved in this way for this reason. And really, what it is a team discussion. It needs to be a discussion of what are the main priorities for conservation? What are we trying to do? What do we need to consider? Education and science, the research and the communication. These are all conversations that need to be had. And so I don't think it's a one there's a direct answer that you should do this. And that's certainly not ever been my approach, because when we were in Goa at Harimau, I also learnt new conservation techniques from our conservator, Mentari Halimun. So I actually learnt new bone cleaning protocols and techniques that I know are safe for paleoproteomics, because they're using even more diluted versions of the stuff that you use to clean the bone surface for paleoproteomics anyway. So use of diluted ethanol and things like that are comparatively safe, because you are as long as you're not dipping the bone in or completely saturating the bone, you're carefully cleaning bone surfaces and things like that. But again, the conversation in terms of the research design and the broader project is important. But some of us also work on assemblages that have been excavated since the 50s, 60s, and 70s. Of course, they're already consolidated, so we need to, or there's other contaminants or chemicals that have been used on them, ones that we have for a long time no longer used, but the bones are still covered in them. So we also need to keep working on techniques to be able to work with the bone that has been treated. So how do we decontaminate bone is important, because we don't want to be digging up more bones when there are bones for us to actually analyze and research further. So I think there's two aspects. One, the discussion and the proper collaboration amongst the team, respect and empathy, and consideration of who is this all for. It includes thinking about the public, it includes thinking about education, and then we come up with a plan together. You come up with a plan together, and then you go with that plan. Sometimes it's going to work, other times it's not, but that's what science is all about. So that's my answer.
Yeah, thank you very much for the insight, and because I experienced also the difficulties during the analysis because the conservation we made before, honestly, and then yeah, I learned a lot, and then we should not we should choose the best way and to have a plan for the future analysis of future study we want to do, and we want to prepare for the collection. Thank you very much. We have one short questions from Ibu Betty Rosi Hermiati. Is there any is there any way to know or to identify about the and to differentiate between the specimen of skeletons that have severe malaria, or only light malaria that can cause the repetition of infection of malaria in one specimen of in bioarchaeological collection in one collection? Do we have any way to differentiate or to identify the level of malaria he got or
No,
infected?
not yet, but that's where the metabolomics would come in, because the more byproduct you're using, the more your body is responding to infection. So that's where metabolomics will be really important, is answering those questions about how the body is actually responding and to the severity of malaria and the expression of malaria in the body. From traditional paleopathology, we can't, because some people will have mild anemia and then have severe bone response. Or some people will have really severe anemia and then have mild bone response. So the relationship between the bone response and the anemia, in general, regardless of what's causing the anemia, whether it's malaria, whether it's thalassemia, whether it's iron deficiency, or all those sort of things, it's really hard to tease out just from traditional paleopathology. We really need to do biomolecular techniques to be able to understand that deeper.
Oh, thank you. And we have, I hope it's answered the question, Bu Betty. Thank you for your questions. And we have one, this is the last question from our participants. You can read it in the chat from Duta. And his question is quite long, the introduction. But this question, it's because of that. Mr. Duta, I would like to ask about the academical debate that is still ongoing on some degree regarding porotic hyperostosis indicating anemia and its relation on the bed, perhaps in malaria-induced anemia case. From your perspective,
From my
please.
perspective, so there was a paper that came out just 2 months ago that said that porotic hyperostosis has absolutely no relation to anemia at all. That research was done on adults, and they were anemia, as you know, the anemic signs develop in children. So there's a big gap in that research. What we have is an extensive amount of clinical evidence that thalassemia causes porotic hyperostosis. Thalassemia is a clear anemia. We have significant clinical evidence to suggest that the porotic hyperostosis is associated with anemia. What anemia? So with thalassemia, we have all these other signs that we look for on X-rays. General anemia, it's just anemia. So if you have porotic hyperostosis or cribrorbitalia, it is anemia. So what is causing it? Whether it's malaria, whether it's deficiency, whether it's B12, you cannot tease that out. You need to really consider the archaeological and biological and environmental context of the site that you're working on. If you are in a site where there are, today it's super hollow endemic, which is the case in West Java. Oh, no, West Java in the past did have a lot of malaria though, so there's a lot of historical evidence. Then you can argue that malaria is probably underlying, because malaria anemia causes folate deficiency, B12 deficiency, iron deficiency, and then infectious anemia. So malaria causes 4 different types of anemia in your body all at the same time. which is why it's such a strong driver. But it doesn't necessarily mean that it is the direct cause of anemia. And that's where people, I think, are getting wrong about it. They're saying that malaria doesn't cause anemia because they're thinking about the direct cause of the anemia itself rather than the underlying or what we call ultimate cause, which is it's driving other things that then drive the anemia. So those kind of things are what's a problem in people understanding it because you need to see it from a system. So the system involves malaria infection, and then it's going to increase the burden in your body of deficiencies that you already have. So if you are vitamin C deficient, you could end up with scurvy. If you've got iron deficiency, you can end up with anemia now. It makes it more extreme. And so it doesn't mean that the malaria is directly causing the anemia, but actually it still is the thing that ends up driving the anemia. And that's something that I am currently in the process of writing a response to this paper that came out that said it's not anemia at all. It's a great paper in terms of they found immune response, but they forget to understand that immune response actually could be driving the anemia as well. And also drive iron deficiency, which then drives, yeah. So yeah, there's a long and very complex system, and trying to tease all of it out is very difficult. And the best that you can do is just very strongly contextualize what you are finding in regards to other pathology you're finding, as well as the archaeology, and the environment.
Thank you very much for the answer. I hope your answer did answer the question from Mr. Duta. And good luck for your study, Mr. Duta. And we arrive to the last occasion, last schedule for today, and we should proceed to the concluding remarks from One. Thank you very much, Dr. Mel. I hope for your beautiful presentation, also beautiful discussion, and yep, One. Maybe I invite you to deliver the concluding remarks for our discussion today.
Okay. Thank you, Mas Ruli, and of course, thank you so much, Mel, for your inspiring lecture this morning. So this study of paleopathology, especially thalassemia and malaria, is still rare in Indonesia and Southeast Asia. On the other hand, this field study is very promising. As we know, we have so many sources on human remains. And as Mel already explained to us, thalassemia and malaria have a coevolutionary relationship in Southeast Asia, with the high prevalence of thalassemia genes believed to provide a degree of protection against malaria. And thalassemia is a blood disorder when hemoglobin synthesis is disrupted. Its prevalence in this region as malaria. And in malaria endemic area, thalassemia carrier have a selective advantage as their abnormal red blood cells are less hospitable to malaria parasite. However, this genetic adaptation can lead to severe thalassemia disease. So this is the relationship as look like a double-edged sword. While the carrier of thalassemia gains some protection from malaria, but the inheritance of two thalassemia genes can result in the severe thalassemia disease. As clinical consideration, this study is very important for health program in this region. Probably in the future we can make like degree by research program, especially for paleopathology on thalassemia and malaria. We can manage together between this institution. And again, I would like to thank Mel and her team, also researcher from both CPIS and Research Center for Archaeometry, also Pak Truman, who realized this collaboration, and also all colleagues who attend this lecture. And again, please keep in touch with us for the next dissemination program. Thank you so much and good afternoon. for everybody.
Thank you very much for the concluding remarks and very very dense, very really like complete and information you gave us. And I hope, as a part member of a team, I hope this collaboration will be like last longer, and then we can like more corroborate ours and strengthen our connection, our collaboration in this topic. And as last information for us, I would like to promote our collaborative works between the UKSW, University in Salatiga, National Research and Innovation Agency, Kementerian Kebudayaan, the Ministry of Culture, Indonesian Heritage Agency, about the forthcoming UISPP Congress that will be held in Salatiga from the end of October until November. And finally, we arrived to our at the end of our talk show. Thank you very much, Dr. Melandri Fogg. I really wish to see you soon and to work together with you. Thank you very much, Pak Sofwan Nurwidi, the head of Research Center for Archaeometry, National Research and Innovation Agency, all the participants here, and also I would like to thanks to Professor Truman Simanjuntak for to make this collaboration happen, and for the great collection on the work of Gua Harimau. Thank you very much, everybody, and see you on the next dissemination. Please follow our updates from the social media of Pusat Riset Archaeometry and Center for Prehistory and Austronesian Studies. Bye-bye. See you next time. Bye. Thank you, Mel. Thank you, Prof. Truman. Thank you, Pak Sofwan. Thank you all. And thank you also. And thank you also for the participant in YouTube. See you again.